Until the early 2000s, floral nectar was largely thought to serve the purpose solely of attracting pollinators and facilitating mutually beneficial insect-plant relationships. However, a growing number of scientists now argue that floral nectar is really more of a manipulant than an attractor. They say that rather than facilitating symmetric insect-to-plant interactions, nectar actually changes insect behavior to make it act more favorably to the plant. Nectar as a manipulant can either act chemically or physically. Some plants use psychoactive compounds, including nicotine, cocaine, atropine, and caffeine to modify insect behaviors. Others physically alter the insect in different ways, such as how the acacia ant plant will modify the digestive tract of an ant to make it dependent on sucrose-free food. Plants can also modify behavior simply through varying the amount, concentration of, and physical sites of their nectar distribution. While not all of these compounds can be considered manipulants necessarily, this video will discuss why plants are now seen as more manipulative than previously thought. I'll discuss nectar, either from floral nectaries or extrafloral nectaries, and its relationship to insect behavior. This hypothesis has ignited debate and forced entomologists and botanists alike to reconsider what the line between manipulative mutualism and symmetric mutualism is. This video will focus on how floral and extrafloral plant nectar plays into this new line of thought. If we're defining relationships as manipulative, it's useful to first define what makes a manipulative relationship. Manipulation is, broadly, one or more species changing the behavior of another species for the benefit of that original species. A manipulant is a substance that modifies the behavior of the insect during or immediately after the consumption of or exposure to that substance. Manipulative adaptive parasitism is common in insect relationships. For example, some ichneummonid wasp larvae force spiders to spin them special cocoons to protect the larvae before completely killing the spider. Aphid parasites, as another example, will mummify the host aphid to use as housing for pupation. These are clear examples of parasitism because the host is clearly harmed or killed over the course of the relationship. However, pollinator-to-plant relationships are not classically parasitic, right? Pollinators receive a clear benefit, nectar, for giving a benefit to the plant, pollination. A textbook example of mutualism. Yet new research caused some scientists to question whether or not this sort of relationship was always a straightly mutualistic relationship, or if it was really a form of manipulative mutualism. Could plants manipulate insects to get a benefit? Let's explore this problem through a discussion on plant nectar. Biernowski and Kartar were among the first to discuss nectar as a manipulant in 2004, in a paper that explained how plants would manipulate the behavior of their pollinators in order to make each insect visit each plant for a shorter amount of time. Plants do this by varying the distribution of nectar within their own inflorescences, which means that insects would receive variable amounts of nectar from flower to flower. This uneven distribution discourages risk-averse pollinators from staying at plants for too long, and allowed more diversity of pollinators, which is good for the distribution of pollen from plants. Graham Pike built on this work and argued that floral nectar should be seen as a manipulant rather than an attractant. He observed insect foraging behavior that suggested insects received information and stimuli from the nectar volume, composition, and concentration that affected their behavioral choices in a way that was beneficial to the host plant. This sort of physical distribution is one form of evolved manipulation of insect behavior. Yet, the physical distribution of nectar is far from the only factor that makes plants manipulative. Chemical manipulation is a fascinating element of this debate. Nectar is composed of sugars, lipids, and proteins, which are termed primary metabolites. Secondary metabolites are also common. These include a huge variety of compounds that are specialized based on the plant, including scent compounds, toxic chemicals, tannins, phenols, alkaloids, and terpenes. It is not surprising that a substance such as cocaine would cause ants that interact with it to be dependent on it. Other substances like caffeine lead to increased speed, precision, and food consumption. Plants have clearly evolved these compounds to be used in order to manipulate and control insect behavior. Yet not in all cases, it's not entirely clear if it is a controlling of the insect behavior or simply a mutualistic interaction. One fascinating secondary metabolite is chitinase, which is found in the nectar of the ant plant, Acacia mimesophyte. In the Acacia mimesophyte, the extrafloral nectaries secrete chitinase, a compound which blocks invertase, an enzyme that hydrolyzes sucrose in ant midguts. This makes ants dependent on sucrose-free acacia plants for nutrition. This specific mechanism stabilizes the mutualistic relationship between the plant and the ant by enhancing the degree of specialization in the ant midgut and tying the performance of the host plant, the acacia, to the survival of the ant population. Heal et al. Studied this mechanisms of specialization by feeding one group of ants regular acacia nectar and another boiled nectar. They found that the group that had been fed the boiled nectar had increased concentrations of invertase, which enabled researchers to hypothesize that the enzymes in the nectar had indeed inhibited the ability of the ant midgut to digest sucrose. Martin Heal reflected on this work and other similar papers in a 2015 review. He raised a question: does manipulation in a mutualistic relationship turn the interaction into parasitism? Heal cites a study in which worker ants that fed from lycaenid caterpillar secretions were significantly more likely than ants that did not feed on their secretions to show aggressive behavior as well as decreased locomotory activity. This is a clear example of an asymmetric dependence. While lycaenid caterpillars need ants in order to survive, the worker ants do not necessarily need the food resources secreted by the caterpillars for nutrition. Still, the secretions enforce cooperative behavior from the ants, leading Hojo to label this interaction as a parasitic one. Heal believes that his work on the acacia-ant relationship reveals a parasitic relationship of the same nature, since the plant is the one in control of the interaction. There is still more research needed to be done to analyze the true benefits of nectar to the ants and other insects who engage in these type of mutualistic relationships. Well, there is this growing line of thought that many relationships are more manipulative than previously thought. It is important to question what the value of this categorization is. Surely, some relationships are more manipulative than other relationships. At what point does a symmetric mutual relationship become a manipulative one? To what extent does free will of insects exist? There's a lot more research to be done on the more philosophical and also more biological side of these questions. For example, there's very little research done on the role of secondary metabolites in extrafloral nectaries like those of the acacia ant plants. But this hypothesis opens up an exciting line of discussion as to what the real nature of mutualistic relationships is. Yet, a more thorough understanding of the actual costs and benefits of plant and insects interacting through nectar is left to be desired. When the insect receives a net negative outcome from the interaction, mutualism becomes parasitism. However, exactly what a net negative outcome would look like is undecided and in flux. Scientists including Kessler, Wright, and Baraki have studied the manipulative effects of caffeine and nicotine in floral nectar, determining that both substances play a highly manipulative role in insect-plant relationships. Yet there's still room for research on many other psychoactive compounds in plants in the context of manipulation. Perhaps this new lens could inspire scientists to study these compounds in a different light. On a closing note, the insect-to-plant relationship is not necessarily a two-way street. Insects are known to introduce microorganisms to plants, and nectar-dwelling microorganisms such as yeasts and bacteria have a significant effect on the nectar chemical profile. It is known that these microorganisms alter the concentration of primary metabolites and can alter the profile of secondary metabolites, but little research has been done to study their role in the manipulative aspects of insect relationships.